Yeah, so today I'm going to be talking to you about what Revel does. We're a designer enzyme engineering company. Peter really kind of teed me up talking about damaged products earlier, so hopefully the breadcrumbs he kind of laid out will lead us to lead this discussion. So I want to start off with just a brief pause and thinking about what is aging. And one of my favorite definitions of aging is this. Aging is the accumulation of damage in molecules, cells, and tissues over a lifetime. And what I mean by damage, is accumulating covalent changes and modifications to long-lived proteins in the body. And accumulation of damage often looks something like this. Over time, there'll be a gradual increase in these changes, with a corresponding increase in the risk of disease. And eventually, the burden of damage becomes so great that the physiological function of these proteins becomes compromised, resulting in disease and eventually death. And to give you an idea of what I mean by damage, I'm showing here the amino acid lysine on the left, and its corresponding damage product, CML, on the right. And so lysine is a component of every protein in the body. But over time, lysine becomes glycated by sugar, forming various advanced glycation end products, one of them carboxymethyllysine. And you might recall CML from the previous talk by Pankaj was highlighted as a toxic AGE modification. And so at Revel, what we do is we develop enzymes to repair this damage from aging at the molecular level. To do this, we use a type of protein called an enzyme. So enzymes are like antibodies in that they can bind to particular molecules, but their defining characteristic is the ability to catalyze chemical reactions. And so at Revel, we apply enzymes to carry out chemical reactions to repair damage from aging, thus reversing aging at the molecular level. We believe the opportunity is indeed massive. So enzyme therapies are a $16 billion market already. These are dominated by successful drugs like Botox. Some of the audience may have partook of this at this point. Enzyme therapy is also another common drug, and these are used for lysosomal storage disorders. But generally, the enzyme therapy market pales in comparison to development of the monoclonal antibody market in the biologic space. At Revel, we have a unique technology to develop new enzyme therapies targeting validated and unexploited hallmarks of aging. We are applying this technology to develop new enzymes that we believe will unlock massive value and expand the enzyme therapy market to diseases of aging. As a first mover in this space, as Frank mentioned, there's many challenges that we face, but you'll see, as the presentation proceeds, you'll see we're making good progress on developing these new therapies. So for the remainder of the talk, I'm going to go through our process for developing these enzymes and how we plan to realize value with them. To start off, we identify validated, unexploited hallmarks of aging and disease, things like carboxymethyllysine and others. We have a nice pipeline of targets of interest that we're pursuing now and plan to pursue after Series A. With a molecular hallmark in mind, we then undertake the endeavor to develop enzymes to repair that particular type of age-related damage. And this is a bit the secret sauce of Revel. Developing these enzymes and biologics is a very challenging process. And so we have an enzyme engineering platform that allows us to develop these new molecules and these new modalities to carry out these specific chemistries required. And then finally, with enzyme in hand, we aim to realize value across biotherapeutic and industrial applications. And I'll go into a little bit more about those other applications later. So I'm going to walk you through each of these steps over the next few slides. To start off, we identify validated unexploited molecular hallmarks. To do this, we look across thousands of metabolites and proteins to try to understand what is changing with time. As Peter mentioned in the last talk, there's certain types of damage that covalently accumulate, and the body has no mechanism for getting rid of. One can think of them as the metabolic waste products that there's no closed loop for. These damaged products include things like calcification, a modification mentioned by elastrin earlier. These include things like CML and glucosylane crosslinks, two modifications being pursued by Revel. They also include intracellular changes like accumulation of lipofuscin. For a given hallmark, we ask several questions. Is there clinical evidence for cause, role, and disease to build confidence around pursuing that hallmark? Is the mechanism of action accepted by scientists and clinicians so that we're not fighting an uphill battle when we develop an enzyme to reverse a particular modification? And third, is there a therapeutic opportunity for an enzyme product? Wrapped into that, have others already pursued this target in the case of calcification? Once we've selected a hallmark, we then develop enzyme treatments to repair that particular type of damage. And this is the most challenging part of our development process, developing enzymes capable of performing the required chemistry to reverse these modifications. These enzymes don't really exist in the human body because with aging, as again mentioned by Peter, these modifications only accumulate late in life, post evolutionary selective pressure. So we have to go into the natural world and look for enzymes that may be able to convert these damaged products to their native physiological form. And to do this, we have our engineering platform. We start out with a computational search to identify potential enzymes that we believe have the required activity to perform this reaction. We then generate billions of enzyme variants that are partially optimized. We select from these enzyme variants for ones with slight improvements and iterate this process using a technique known as directed evolution. Directed evolution was awarded the Nobel Prize a few years back. It was awarded to Francis Arnold and several others. And it's a technique that's been used for decades to evolve many enzymes used in laundry detergents and other applications. And using this technique, we eventually narrow down to tens of candidate enzymes that perform the desired chemistry to the degree we require, and then can use those enzymes in our applications. And I want to pause here a little bit and just talk about our enzyme engineering platform. It has many advantages to potential competitors. We use a genetic selection technology that allows us to assess millions or billions of enzyme variants on a single Petri dish. By having such rapid experimental identification, we can scan through hundreds of millions of variants a week for relatively trivial cost. This is in contrast to companies like Novozymes, a European company, and Codexis, based in San Francisco, both major players in the enzyme therapy and enzyme industrial space. These companies use a variant library and plate screening and heavily roboticized solutions, which at the face of it look efficient, but are in fact very time and capital intensive, limiting them to just a few 100,000 variants a week. And so what this boils down to is at Revel, we're able to scan through 1,000 times more variants at effectively less than 1% of the cost of some of these other methodologies used by companies in our space. And to give you a little bit of a taste of how this looks in action, I'll give a little example with developing an enzyme to repair CML damage. So as may be mentioned by Pankaj and others, CML is a toxic aging modification. It accumulates covalently over time, and there's no enzymes known to perform this reaction. We're very interested in CML, both as an aging modification and for the role it plays in diseases such as diabetic complications like diabetic retinopathy, among others. To develop an enzyme capable of reversing CML, we applied our enzyme engineering methodology, starting with a computational search to identify initial enzymes with potential activity. Here I'm showing a crystal structure of one of the enzymes that we identified. This is just a molecular representation of what that molecule looks like. We then created billions of enzymes with partially optimized activity. To do this, we selected a set of sites within the starting point enzyme to randomize. And so this is effectively a optimization problem that requires a way to sort through these variants for the ones that are improved. In this instance, we were able to identify enzymes with improved activity from about a 1 billion member library in just one round of engineering. And the data generated from this simply shows that the engineered enzyme has significant activity compared to the starting point enzyme. So in black is our starting point. It has activity that we theorized that could be converted to working on CML. And no matter how much substrate you jam into that enzyme, it's not active. Whereas the engineered enzyme we developed has significant activity that brings us much closer to where we need to be for therapeutic application. And this really just serves to highlight the power of our platform. We're applying this system to many other of the modifications we're currently pursuing and plan to pursue after Series A as well. So I've talked to you a little bit about steps 1 and 2. The third step is how do we realize value from all this cool science? And that's across biotherapeutic, diagnostic, and industrial applications. What we're effectively doing is creating a portfolio of engineered enzymes capable of performing chemistry that no one else has. This is in contrast to many of the other enzyme engineering companies out there where they're working on enzymes that other companies can access. They're naturally occurring. These enzymes have been highly engineered, and so the chemistries that we're accessing are effectively brand new. For each enzyme that we develop, we believe there's very valuable applications in the biotherapeutic space. The reason Revel exists, of course. The total addressable market for just 2 of the indications we're pursuing, diabetic retinopathy and rheumatoid arthritis, is on the order of $20 billion. Potential partners we're also in contact with include folks like Calico, who are interested in the technology platform, as well as folks from BMS and AbbVie. In addition to the biotherapeutic applications, we believe there are low-risk, early revenue opportunities in the diagnostic and industrial enzyme spaces. And the justification for this is with an enzyme known as asparaginase. It's a first-line treatment for childhood leukemia. And asparaginase, while also a biotherapeutic, is used in the food industry to remove acrylamide from your fried foods, hopefully. So that's a little bit about what we're doing and how we're doing it. The competitive advantage that we see, in contrast to some of the other players in the space, is that many of these other companies are competing in very crowded markets focused on similar enzymes and applications. Just a few of these companies, Novozymes, Codexis, and BioMarin, have a combined market cap of over $30 billion, but they're very focused on similar applications. There's significant redundant focus on enzyme replacement therapies, a space that will soon face competition from gene therapies. And there's also limited market expansion opportunity for things like asparaginase and collagenase enzymes, which have been around for decades. There's fundamentally a lack of innovation in new enzyme therapies compared to what we see in the monoclonal antibody space. And so at Revel, we are developing differentiated products targeted to new markets. We believe by applying these enzymes both to targets in aging and using those chemistries to develop applications in the industrial and diagnostic spaces, we'll really differentiate ourselves and stand apart from some of these other companies. To date, we've raised $8.2 million with support of folks Kizu, Starbloom, who I believe the representative is here, and as well as Formic Ventures. That money is being spent to put together a Series A data package focused on the CML oxidase enzyme, which is being developed for application in diabetic retinopathy. We're also developing a citronellase enzyme, which has application in rheumatoid arthritis, as well as cross-link breakers targeting glucosylpane cross-links, and finally, some undisclosed applications we're pursuing. We're anticipating raising on the order of $20 million Series A in the first quarter of 2024, and that money will be spent to fund our clinical development activities, as well as expand our pipeline. There's a lot of interesting aging modifications that accumulate with aging, and we are excited to dig our hands into those as soon as we have the team and the resources to do so. And so the team is led by myself. I'm a Stanford-trained PhD. Prior to Revel, I was at Google X briefly and spent a little bit of time in biotech consulting. Dr. Kevin Desai is also here. He's the head of protein engineering, and he's responsible for significant development of the enzyme engineering technology that we've been pioneering. We also have a stellar team of advisors and board members. And I'd just like to highlight, for some of you who have been following the advanced glycation end product space for a long time, you may recognize Ken Mock was the former CEO of Alteon, the company that in the early 90s was trying to develop small molecule drugs to target some of these modifications. All of these bios are available on our website. And finally, I'll just close where I began. So if aging is accumulation of damage in molecule cells and tissues, then my vision of rejuvenation is repairing this accumulated damage. That's the mission that motivates me, the team at Revel, and I do hope you'll join us. Thanks very much.
Thank you. Fantastic presentation. Actually, the first one where I saw revenues on the slide.
Yeah.
So do we have questions in the audience? Here's one. In the first row here. Here, no, just here. Yeah.
Yeah, nice presentation. I just want to know, how do you deliver these enzymes to in your body? And do you get any immune response? You know, it's a synthetic kind of enzyme, right?
Yep. So 2 questions. How will we deliver them? It's dependent on the application. So for diabetic retinopathy, the current standard of care is with anti-VEGF antibodies, and those are delivered intraocular, via intraocular injection. So that's the route we pursue for that. For rheumatoid arthritis, the delivery is currently, standard of care is Humira for the most part, and that's delivered subcutaneously. We're most likely pursuing a subcutaneous administration for that as well. And then for some of the other applications, that's TBD. Yeah. The second question, can you repeat that?
I was just thinking about the immune response. You know, if you do multiple injections of this, something new in the body.
Yep, absolutely. So all biologics are immunogenic. We're relying on the fact that our treatments will be acute versus chronic. Humira is a chronic treatment for rheumatoid arthritis because it needs to constantly sequester TNF-alpha. The modifications we're going after for rheumatoid arthritis, these citrulline-nated proteins that are one of the drivers of rheumatoid arthritis, those accumulate over many years. And so if we can clear them out, we can remove that driver of inflammation, and it can be an acute treatment, which has a less of an issue with immunogenicity.
A quick question on just enzymes as a therapeutic. Are you worried about, or how are you screening out and overcoming, the degradation and poor biodistribution effect of an enzyme? Because they're highly reactive, they're prone to being broken down, so they often need a higher level, which could lead to toxicity, and distribution is a bit lower. So how are you attending to that?
Yeah, for any biologic, the biodistribution is a key question. These molecules have not been in animals yet. Based on data we have from comparable drugs, the biodistribution will be a thing we have to address. The key difference with antibody drugs, though, is the enzymes are catalytic. So an enzyme can do its job over and over. And so the dosing profile should be much lower than you'd have to dose with like a biologic drug, because we're not trying to sequester a target, we're just trying to remove it. And the enzyme can do that job many times.
Okay, there's one more.
How do you engineer the enzymes for specificity? In other words, make sure you don't have off-target effects.
Yep, actually I removed that data from the slides, but so in the case of the CML example, by evolving activity towards a particular substrate, we often eliminate activity from other substrates just by virtue of narrowing the specificity. It's akin to a low affinity antibody might have many targets, but if you make a high affinity antibody against one target, it will probably be very specific. And so that same sort of effect occurs with us. And we've already seen, and one of the enzymes we've engineered had a very high level of off-target activity on a serum protein or a serum molecule. Once we engineered it, that activity was gone.
Okay, thank you. Brilliant talk. Thanks, Aaron.
Thanks very much.